Hi guys. Um, what a day. Sorry, for some reason, everything feels like it's going wrong today. My internet is acting up. My husband is building things with power tools outside the window. It's hot. It's day 3. I just messaged Dr. Tudge. I had given him the time 12:30, so that's on me. I'm not sure if he'll be able to come a little early. If not, maybe someone else will pop in with me to fill the time, and we can talk. So it's not just me the whole time. I'm getting very camera weary. I still have to do the interview with Carol later in the day. So yeah, I can't handle any criticism. Don't give me any criticism now. I can't look. I'll start to cry. It's been that kind of day, most likely from lack of sleep and stressing and everything going wrong today. Trying to stay ahead of it, but thanks, Moa. I'll put on my introduction voice, which everyone loves. I can't. You don't want to hear my cranky mom voice, or my nagging wife voice, or my scolding the dog voice. But if anyone else wants to rescue me and talk with me, I think Darcy's busy rescuing me in the shells talk, and Stacy's busy rescuing everything in the Chia Xuan Shu talk. So Rita's going up at noon. The good news is you can watch Rita's talk at noon. It's very short. I think it's maybe 8 minutes. Even with the introduction, I think it's that short. So hi, Tammy. So feel free to pop out and watch Rita's. Dr. Tudge, if he does come early, which I'm not sure if he will, since I told him 12:30, will completely understand, and we can hold it. What I did was I backed up this session to noon to give us a full hour before Tammy's presentation, because that was so incredible. I knew we would all want to see that, and I thought a half hour wouldn't be enough time for us to cover everything we needed to cover with Dr. Tudge. So I backed it up. I forgot to confirm that with him, the most important person in the room for that talk. So he's planning to be here 12:30. I know that for sure. I can just check my email quick and see what he said. Yeah, yeah, Kelsey, I just knew it was going too well on day 1 and day 2 for it to hold. But fortunately, if we start strong, you guys will remember that. And we'll finish with Carol, and how can that, Tammy's talk and Carol, they're going to carry us through after this morning. We are doing great. I can't even fathom. I haven't been able to enjoy it from an attendee's point of view quite the same way as most of you. But yeah, even from a hectic organizer point of view, it's been pretty incredible. Yeah, 2 months to plan, and then we wouldn't have been able to do it without you guys, Kelsey, and everybody else who put together on a moment's notice their awesome presentations. Once we knew we weren't going to be in person, and we did, in person we had 1 day, and every hour that was 8 talks. And then when we went virtual and we suddenly had more time, I thought, well, let's cram a bunch more talks in there. So we scoured the internet and found the experts, and found, the people on the moderation team who were willing to help. And yeah, I still kind of boggles my mind, and I was on the back end doing a good deal of it. So thank you, Hop-In, if you're listening. I don't know if they monitor things. I can't imagine they would, but maybe if they see it in the playbacks. Thanks, Colleen. Yeah, it's really blown up a lot of things in a good way. There's a person who just joined Shenzhen from Taiwan, who just joined on the 9th of this month, 2 days, 3 days ago, who has had tremendous success breeding hermit crab Zoe in even Clypeatus, which I think it's a she, got way back when they could get them. I think she said 11 years ago. Yeah, that Crisell, I know, that giant thing with the spray bar and then the land tank, that would really take it to the next level, like commercially or however we want to think of it, breeder wise. Because that's incredible. Think about how many would survive with that volume of water and the ability for them to move about. Yeah, probably $5,000. That's a little out of my budget, and I think my space range too. I've asked quite a bit of my husband in terms of space. I keep expanding, and so we'll see. Yeah, if I get $1 million grant from some, research organization, or better yet, they do it and I'll consult. I'll come and, oversee if they need it. That, people aren't going to need me or others like us for long, especially with us being so generous about sharing our methods and everything. I believe in open source and, yay, Stacy! I'm glad you're here. Can you come on screen, Stacy, and or are you screened out too? I'm just feeling.
I'm here.
Yay! Oh, here's Dr. I'm so sorry.
That's okay. I had it written down somewhere as 12:30, and I don't know who was. I never changed it, so maybe whatever happened.
It was me, and you confirmed with me a few days ago, and I said, yeah. And then when I got Tammy's talk on Thursday, and that was at 1, and that only gave us a half hour, I went in and backed it up to 12. Probably thought I told you, or completely spaced.
Anyway, we have time.
But bless you for coming in. You guys, if you double click on his picture, he'll be big and I'll be small. Maybe I'll just sort of stay in the background and help you with questions. Like, would that be good? Or do you want me to totally disappear? I am good either way.
I don't mind. You can stay there. Are they going to be sending questions again? Chat and stuff. Okay. All right. That's fine. So people don't want to be seen because they're eating and probably sitting in their pajamas again. So I guess that's
yeah. And I can pop out too. I'm more than happy to pop out and give you the stage. You are lagging just a little bit for me. Is anybody else getting a lag with Dr. Tudge? Say something, Chris, and we'll see if you.
Okay. Hi there, everybody. Most people are saying no lag, so it might just be you. I think we had this problem again last time, Mary, that we chatted. So.
Okay, that's excellent. I mean, it's not excellent, it's excellent, but nobody else. I am having a tremendous lag, so I'm going to disappear and see if that helps me see you better. And if you need me, just holler. I can pop back on in a moment's notice.
Okay.
Thank you for coming early. Sorry about that.
So just before you go, so we've got until 1.
Well, we have until 1 because I think we all want to see Tammy's presentation.
Yes.
If we pause it and need to come back, that's absolutely fine. I can extend the session. We can decide that at 12:50 or whatever and see where we are. Okay. Thank you.
We'll see what we're going to do, what we get through in 50 minutes. So. Okay. Well, thank you everyone again for joining us and joining me on this. We covered a lot of what I wanted to talk about today in the question time after my presentation yesterday, but I just wanted to reiterate what the idea of this little session was, and that was to talk about the types of data collection that you guys can do to generate good data that could be used for research and for publications. There are so many of you, you have great access to your crabs, and many of you are already collecting probably the data that's required. But I just wanted to talk to you a little bit about how that data should be collected and the veracity of it. If we're going to use it for publication, then you have to make sure that you cover all the bases and have things like controls covered and good authentication of the data is basically what I wanted to talk about. And some of you might already be doing it, and some of you may be collecting data but not doing it at a level that's suitable for this. So I was thinking about what we chatted about yesterday, and I came up with 4 topics that I thought we mentioned where data that you guys have could be useful for getting at some of these questions. And just very quickly, they were sex change or intersex individuals, hybridization, individual identification, and Moa, you have great data on that right now. And also the idea about the function of sexual tubes. And there were 4 things that I remembered that people brought up yesterday, and we did talk about what we could possibly, how we could get this data a little better. And I talked about saving specimens, photographing molts, saving deaths, saving instars if you are breeding them, and keeping them, and they'll basically last forever in alcohol, and they could be used at some point in the future. Photographing and videoing stuff, but making sure that you do things like if you're photographing, there needs to be a scale, there needs to be a date and a time, and if you're videoing, the same sort of thing. And then many of these research questions depend on being able to identify individuals. The sex change and possibly even the sexual tube stuff may require that. So that's where I wanted to start off the conversation. And I thought perhaps some of you might have some questions about data you already have and what you may be able to use it for, or you wanted to start collecting data and how you wanted to go about doing that. So, and if you have any other topics to add to those 4 that I brought up, of course, I'd be more than happy to chat about them. So let's start about that. Okay, Mary, can you repeat the process of saving specimens? Okay, excellent. I mentioned before the preservative of choice for any scientist or taxonomist is 70% ethyl alcohol. And that's good for molecular work as well. If you want to do some genetic analysis later, extract genes or proteins or whatever, then that is also good for that. And basically, if you've got specimens sitting in that, they will last for years and years and years. And in fact, there's stuff on the Smithsonian shelves that's been there for 100 years in ethyl alcohol. The older it gets, the harder it is to extract good genetic data. But basically, for our time period, for the next decade, anything in alcohol is going to be great. No, Robin, not formaldehyde. Formaldehyde is no good for doing genetic extraction. Formaldehyde is good for preserving color and keeping specimens soft and supple. Alcohol tends to dry them out, make them a little brittle, but we're talking about multiple decades over that time. So something in alcohol after 20, 30 or 40 years may become brittle. The problem with formaldehyde, of course, is that it is a carcinogen. And although ethyl alcohol is not something you can drink, it's way safer than formaldehyde. And as I said the other day, rubbing alcohol is 70%. I think it's isopropyl alcohol is what it's called, and it's good enough for this job as well. So you could buy a pint of that from CVS, and if you use it in small amounts for small specimens, and store them in glass jars that have good sealable lids, effectively they will last forever. I would recommend you label stuff if you put it in there. Lead pencil on a stiff white card is best. Don't use inks. Most inks run in alcohol. And so you might want to put in there, the date the specimen was, if you have some sort of identification for it, name or number or whatever, and the reason why you pickled it, etc. Perhaps some other data on who the crab is, how old they are, etc. I'm sure you have records for it elsewhere. That could all go onto a card in there And then in 5 years' time, if you go back to your shelves, you'll still have that data available to you. So that's what I would recommend. And if you're pickling most specimens, you need about a 20-to-1 volume of alcohol to mass of your crab. So if you have a really big crab, like a large, let's say a small clementine-sized hermit crab that you're going to preserve the whole thing, you're probably going to need half a pint of alcohol to do that effectively. But if you're pickling zoeas and norplii and all these sorts of things, your larval stuff, you really only need a few mils of it to have those specimens in there. But a good 10 to 20 times volume is a good ratio, so that you get good fixation from it. And yes, preserve as soon after death as possible. As soon as something dies, their tissue starts to degrade. DNA really only lasts minutes before it starts to degrade. And so the best thing to do is to get it in there as soon after death as possible. If you know something's going to die and you want to pickle it before its natural death, rather than throwing it in the alcohol, which can really shock it, and they will often throw their limbs when you do that, you'd place them in the freezer for 20 minutes. That cold shocks them to the point where their nervous system shuts down, and then you'll be able to drop them into ethyl alcohol. You don't really want to freeze them solid, but you want to leave them in there for 20 minutes until they become totally immobile and their nervous system has basically it's inactive. It's not frozen, it's inactive. And then you can pop them into the alcohol. And as soon as the specimen hits that alcohol, the penetration into the tissue is fairly fast. It takes just minutes. As I said, if you have a large crab, several inches across, it could take an hour or so for that alcohol to make its way all the way into the middle of that large crab. Smaller specimens, it's faster. And so that's the preservative of choice. And like I said, a good sealing glass jar. They will evaporate over time if you don't have a good seal on them. Any sort of pickling jars and any of those jars that you would pickle or can in are really good. The problem is getting them small enough to keep specimens in. And if you're dealing with larvae, of course, you want a really tiny jar, which we have lots of them in science, but we don't tend to have so much in the kitchen. You really only want something that's going to hold 5 or 10 mils of liquid, and there's not many of them around, although you may be able to find some available for things like people who do oils or beads often keep them in a little tiny glass jar. Anyone's doing therapeutics or herbal stuff, you may be able to buy these little tiny jars that just hold a couple of mil. They have to have a wide enough mouth on them that you can put a specimen in and pull it out. So don't get the ones that have really restricted mouths on them that you could drop a crab through, but you could never get it back out of. I see Kelsey's asking something here about good method of preserving Zoea megalopia on a mounted slide. You can make permanent slides after they're dead and pickled. I would pickle them first in alcohol, then you could mount them onto a slide, and then you can make a semi-permanent mounted slide by sealing it with clear nail varnish. You could put them into any sort of distilled water or any liquid. It's not recommended to put them in the alcohol under the slide because it will evaporate fairly quickly. So basically, take them out of the alcohol, give them a wash in, say, distilled water, put them in a drop of distilled water, make your slide, have enough liquid that the cover slip is not crushing the specimen, and then you can use a clear nail varnish to seal around the outside. And as long as it's completely sealed, that'll be good for quite a few years. There are better media for keeping slides forever. Slides we keep at the Smithsonian are in a special balsam media. That's basically a clear epoxy, essentially, that sets them underneath the cover slip. But that's not something you guys are going to have access to. All right, I'm starting to lose track of what's going on here. Let me just slide back a little bit and see some of the things that were said here. I have to go back to, where was I? No lag. Okay, that's about it then. Brooke, I'm glad that somebody enjoyed the sex of the show. Maybe this is what you want to do, record that program and show it to all of your crabs to get them to make a little bit of crab porn. What was the metric? Robin, what were you asking about preferably metric? Can you pop that back up again? Yeah, you're going to have to watch the replay, Kelsey. Sorry about that. All right, Brooke, here we go. Okay, so theory about shells that some crabs won't grow at a faster rate if they don't see that there are enough resources. Oh, that's interesting. Currently testing this. So the way you would test this, I guess, would be to have tanks with resources and tanks without and document same aged crabs over time. You'd have to make sure all other variables were the same and that the only variable was whatever resources you were testing. And don't do too many resources at once, because if you're depriving them of, say, water, food, shelter, shells, etc., all at once, you don't really know which one of those or all of them is impacting them. So you might think about if you use the same aged crabs, approximately the same size, and probably hopefully from the same cohort, you may be able to deprive them of resources like shell availability and see if that makes a difference, and then try them on another resource like shelter and see if that makes a difference. Because testing too many variables at once makes it difficult to parse out which one's actually making the impact. So that's what I would suggest there. Try one resource at a time. Which resources you think are going to be the most impactful first. Some of them, of course, are dangerous. If you try depriving them of water and food, then you're going to impact their health a lot. But you could just try providing less or more of those resources, like food and water. I'd hate for you to kill your crabs because you are depriving them of something they really need. But you could definitely try things like shell availability. Don't give them any shells in one place and another. And I would suspect that if there's no big shells to move into, a crab's not going to molt. Once it reaches the size that fits the shell it's in, it will hold that size until it has a bigger house to move into. So that's probably an easy one to test. And I bet there's possibly some published resources on that from the wild as well. But having them in captivity is going to be a way better way to test that. And Brooke, other than things like food and water, what other resources you might possibly be thinking about there? Send me another message on that. Risky, yeah, talking about decomposition, basically within seconds to minutes, depending on the size of the specimen. Mary, how can we best help you? Well, really, what we're missing for the sexual tube stuff is actual functionality on live animals. So images, photographs, or videos of sex tubes in action. I'm pretty sure these sex tubes are only mobile to the extent that the coxal segment is. So if they move the leg, they move the sexual tube. They should not have any flexibility or mobility because they're calcified. We have no evidence of that. I've only seen them dead. So anybody recording video that shows that they are basically immobile and not flexible, but they would be able to move with the moving of those two coxal segments at the base. Different species, which is interesting. There's only a few that have the sexual tube like Pilatus. A lot of them don't. And there is a great Nakasone paper that has, I think, about 8 species there with the sexual tubes published in it. And most of them look like the Clypeatus ones, which are just symmetrical, small, flat coxal plates with little bumps on them. Seeing how those crabs transfer sperm mass and reproduce either through video or still photography versus Pilatus that has it, and see if there's any difference, that would be useful. Once again, if it's possible, I know this could be difficult, getting some idea of scale is useful. One thing you could do is you could, if you've got footage of a crab, then later after you've taken the footage, you get a shield or carapace length measurement for that crab, or if possible, turning it over and get a rough estimate of, say, the size of the coxal segment, and then you could record that with it. So when you see the video, someone might want to say, well, how big is this really? And you could say, well, that coxal segment is 2 millimeters across or something like that. You won't be able to do that while you're filming, but you may be able to do that afterwards. So that's what I'm thinking about for sex tubes. There's not going to be a lot of utility in repeating the work that I did on Clypeatus and Perlatus for all species of Cenobita. For example, if people have rugosus and what else is there, purpureus and variabilis, etc. If they don't, only if they have structures that look really different. So I wouldn't want to go around cutting up crabs and that have very similar sex tubes to do all of that microscopy, because I think they're all going to be pretty much the same. But if there are species with sexual tubes that are very different, long paired ones, for example, if there's anything that has sexual tubes like Perlatus, but on both sides, that would be interesting. But I don't think there's a great utility in cutting up every species of Cenobitid hermit crab. I think we're going to find that they're all built exactly the same. We just need to know what the functionality of it is now. And as I said the other day, I would really love to be able to track the development of the sexual tube from juvenile to adult. And that may be able to be done through molts, if it's possible to steal molts, or to rescue at least the part with the sexual tube on it. As a juvenile, by definition, the male should not have a sexual tube. And as soon as they molt to a young adult, my guess is they're going to have a little one, and every molt after that, it's going to get bigger. And then that ties into the data that I had the other day on allometric growth of all those dead ones that we got out of the museum. So even if you can handle your crabs enough to measure a sexual tube, then if you've got crabs that you're keeping for many years that are growing progressively, it'd be interesting to plot the type of work that I did. Carapace length, you could do that with a caliper or a good ruler. Propotus length of whichever leg you want. You could choose any one of those leg segments. We just chose the Propotus of P3 because it was an easy one to measure. And then sexual tube length, you could plot all that data and keep it over years, and you should be able to repeat the same sort of growth data that I got for that one, for that Perlatus one. It might be interesting to plot that for both left and right coxy of symmetrical species. So things like Clypeatus, or what else is symmetrical? I think rugosus might be, if I'm remembering correctly. So a few of those species, simply measuring those structures over time. So for example, you could measure it after every molt of a crab, over time, over years of a male, and plot the increase in body size against the increase in sexual tube size. And it would be interesting to see if there's the same asymmetrical growth or positive allometric growth in other species other than Perlatus. And you should be able to do that without terminating any of your crabs. You should be able to handle them enough and carefully enough to be able to take all three of those measurements on a regular basis. And I don't think it's going to make a difference to do it in between molts, because they're really not going to grow that much. So it'd have to be, post every molt, you collected that data on a particular specimen and were able to follow that specimen through its entire life. So some lifetime growth data would be amazing to get from basically from hatching to a natural death. That information we cannot get from the wild. So there's a project for one of you that's young enough to follow your crabs through. I know some of these crabs can live for 30 years or more. So start young, follow them through, collect that data, and keep a record of it. Back it up, share it with somebody, put it on the cloud, whatever it is you're thinking of doing. Okay. Kelsey, I see 10 minutes ago you asked something, how long to keep them in the alcohol before removing. What do you mean by removing them? They stay in the alcohol. Once you've pickled them, you keep them in there. Unless you're talking about slides, of course. All right. So Moa, back to your individual identification stuff here. What are stitch marks? Tell me what that is. I don't know what they are. I know what cetal patterns are. Where are those cetal patterns? Are you looking at? I think I missed your talk, didn't I? So they are on the claws, or they're on legs, or both. All right, Mo, it might be something for you and I to exchange emails over. I'd love to see some of these images if you've got of them. I don't know what stitch marks are, so you could explain that to me as well. And if you've got multiple species, it would be really interesting to see the type of data you're collecting for both of them. You could easily just flip me an image on email. What I would expect you to be able to do here is to be able to track an individual, prove it's the same individual, okay? For example, if you have it, how would you do that in a tank with multiple hermit crabs in it? You have to have some identifying feature that you can recognize, that you can show someone else, essentially. So let's say it's got, one of its dactyls is broken off and it never grows it back, or it's a one-eyed one, or I don't know if you can't really use shells because they're going to swap them. This is where I've had issues with identifying people who've said they've had the same crab that's changed sex. You have to have some identifying feature of that crab to prove to somebody else other than yourself. I know you all know your crabs well enough to identify them by sight, but you need to be able to quantify what that is. And so then you need to show that the seedal patterns are the same over molts over time, is what you need to do for that. And then if you have a series of images of the same seedal patterns with either no or minor variation in the same individual over multiple molts, and then you'll be able to prove that this pattern is a useful dynamic for a useful characteristic for identifying crabs, even if they change shells, even if you come back a year later and find it in a tank full of 30 crabs, et cetera, et cetera. But I can definitely look at your data, Moa, and we could figure out whether this is something that can make it into a scientific paper or not. I'd be happy to work with you on that, see what you've got. Because it really is quite interesting. If you can, if this becomes a good method for identifying individuals, then the whole community can use it. And then that makes all this other research so much easier if you can do individual identification. So share a little data with me, Moa, and we'll talk about it. Oh, interesting. Risky, you're asking the $1 million question, how to count a crab's age. You guys know, because if you've hatched them like Mary, then you know how old they are. But anything that you picked up elsewhere, it is going to be difficult. Because crabs don't have a linear growth model, but a stepwise one, and they don't necessarily have to molt unless they're physiologically able to molt, there's not a direct relationship between time or size, crab's age. This has been a problem for crustacean fisheries for years, especially when fisheries are based on size. So if you're catching, stone crabs from Florida, or Dungeness crabs from Western Australia, or not Western Australia, Washington State, sorry, or any crab. If you have a crab industry, you often have legal limits and sizes, and those legal sizes are an attempt to maintain a sustainable population, and therefore they're usually a size that they think that an individual is past its first reproductive age, therefore has contributed to the population before you remove it. But there's not a direct relationship between size and age. They haven't found that in any crustacean yet. You could have two crabs the same age of very different sizes, and it all depends on what environmental conditions they grew up under and whether they've been able to molt as many times and grow as many times as they should have. And so two individuals of the same age could be very different sizes. There's a couple of methods that have been used. None of them are very field friendly. They're all lab based. They looked at the accumulation of a particular brain protein called fuchsin, it was called, and that accumulated in the brains of commercial crabs, and by sectioning the brains you could tell via accumulation how old they were. But you can't use that in the field. You can't take the brain out of every crab. They were trying to correlate it with size and things. That is the $1 million question, Risky. And no, there's not a good way to tell. Only if you've had that crab from the beginning, or somebody's reliably had it from the beginning and gave it to you and can say this is 10 years old and you can keep counting after that. There is no good way to identify them as far as I know. You can't even collect molts or count how many times they're molted, because they don't molt on a regular basis. They don't molt like once a year, once a month, or once a week. They molt when they're physiologically ready, when the environment is good for them to grow. So that would be interesting, but I don't think there's any way we can do that right now. All right, so this would just be by the size of the shell, Brooke. Thank you for getting back to me on that. So if there are only shells that are the exact size they are, or a little bigger, and they don't see there enough for them to grow, because we want to know if they cause them to grow at a slower rate. That's doable. That really is. You'd have to be careful about how you measure size of the shell. You'd have to look at whether it's the opening, the operculum of the shell is important, or whether it's the actual mass or volume of the shell, and you should be recording that data. You might also think about different shaped shells. As you know, there are many different types of shells: long spiral, short spiral, flat spiral, some with round openings, some with rectangular openings. So you'd have to control for that. So you'd have to choose a shell type that you know they use maybe the same shell type of different sizes, measure the openings, and then change just those shells and provide them in either smaller, same size, or larger proportions and in numbers, and see if you see a change in growth rate. This is going to be a long-term thing, because the best data you're going to get is more data points, the better it's going to be. So this is not sort of thing you can do for a week or a month. This might be a thing you need to do over a year or years to collect this sort of data. So control for type of shell and shell size. I think the opercular opening of the shell is going to be the most important, because they have to fit themselves into it. But remember, there's many different types of shell architecture. Some of them are heavy, some of them are light. And so you're going to have to control for that. And the best control would be to use the exact same species of shell, different sizes. So you'd have to collect a whole bunch of them and make sure they're all similar. For example, somebody told me yesterday in the talk that crabs like all the manky shells with all the bits knocked off them and scraped and things accumulated on them, rather than nice, flashy, new, shiny shells. So you're going to have to try and control for that too, because you don't want a crab selecting a shell because it prefers the coating it's got on it rather than the size. So cleanish shells of the same species of different sizes will be the best way to gather that data. And once again, Brooke, if you want to talk to me about that, we can talk about that over email. We can lay out a research plan and control for things and talk about that. So Mary's just popped one to the top there. We don't yet suspect hybridization, but we have seen some strange captive bred behavior. Okay, let me read the little more of that. Exhibited odd behavior that seem more like behaviors of compressors, but they aren't growing in size. So from reading this, their different species have different behaviors. That's something I did not know, and you guys know better than I do. Behavior is a tough thing to control for, though. The only way to unequivocally prove hybridization would be through comparison of genetics and gene sequences. That's the only way to definitely check for that. Unless you've got really distinct morphological characteristics of two species. Let's say it's color, one's always brown, one's always purple, and then you end up with some sort of purple-brown mix. That's a good indication of hybridization, but I would back that up with genetic data for sure. Yeah, that would be the only way to look at that. And we can do genetic analysis of individuals all the way from post-hatching all the way through to juveniles and adults. So it doesn't necessarily have to be wait until they're adult to do the genetics. So if you suspect hybridization and you collect, 10 larvae and put them in a container, we may be able to sequence that sort of larvae. I have access to a geneticist at school who may be interested in a project like this. As long as the data was collected with great veracity, so there was no chance of it being all mixed up, then we would probably be interested in doing some of the sequencing. and see how that goes. I could definitely interest a student in that if we had the data. But you guys want to think about what sorts of questions you want to answer about hybridization. So do you want to know they're hybridizing? Do you want to hybridize? Do you want to know if they're capable of hybridization? Do you want to make hybrids? Is that what one of the goals is? Or do you want to not have hybridization so that you want to keep species separate and clean? So think about what your goals are and what information you need to know from that before we go into that. Okay, Moa, thank you. Just posted something up here. Presentations will be available to watch again soon. Okay, thank you. Ridge, elongated bumps up in their large pincer. I know what the stitch marks are. That's probably what they call their stridulating organ. They actually make noises. I presume you guys know that. They chirp, and they do that by rubbing, I think it's one of their legs against that stridulating organ. So I think that's what those stitch marks are. A ridge of elongated bumps, yep, that look like those parallel lines. I think that's what they are. These are probably making those noises to communicate with one another. With all of their legs on the ground and their shell on the ground, if they stridulate, they probably pass that sound through the substrate to other crabs. They could be intimidating one another with it. They could be attracting one another with it. They could be talking to one another with it. Those stitch marks are probably what that stridulating organ is probably what that stitch mark is. And I would hope some of you guys have recorded stridulating behavior, maybe got sounds of it. Do you all know your crabs make sound? I hope you do, and I hope you enjoy it. So risky, I'm just working my way down some of these here. The big brevimanus in the wild tend to have tight-fitting pants. So what would that mean to Brooks' theory? That's most likely the bigger they get, the less shells that are available to them. And so they are restricted. The shells become a restricting resource. In many places, the larger you get, there's not a lot of shells available. They don't wash up as frequently. And you have to, these crabs are not going into the ocean and pulling these shells out for themselves. And also, of course, bigger shells get collected by humans. So in any environment, big shells are not going to be available because people are picking them up. So shells become a limiting resource. That is the reason why the coconut crab doesn't use the shell. It's got so big that there are no there's no consistent supply of big shells available for them to put their butts in. So the bigger they get and the harder they get, the less they need to use the shell, and they then can release themselves from that resource limitation. So coconut crabs use shells when they're juveniles. They bring them out of the sea with them, but once they've gone through a couple of molts, they dump the shell and become a little baby, and they hide out until they're hard enough and big enough to care for themselves. But big land hermit crabs will have trouble finding a consistent supply of different shells. So that's why they would stick with something they had and might limit their growth and molting because they don't have the availability of shells. But that's something you could test. Get a big one, get a selection of ever increasing shell sizes, keep offering a slightly larger shell to your brevimanus and watch them grow and fit the shells you give them. They have indeterminate growth. They will continue to grow forever within the limits of physics and engineering. And so and of course, as they get older, they may also lose some of their physiological fitness. So yeah, that would be interesting to see that and to check out those shells. Oh, that's really interesting. You have Mo. I'm just reading your thing from 15 minutes ago. Posit the somatophore over the edge of the shell. This is what I always suspected, but I've never seen it. That's the only thing I could think of is why you would want a tube like that. As I said in my talk, it becomes less risky. You have to expose less of yourself out of the shell to do that mating. It would be nice to have documentation of that, and I would be really interested in any images or video you have of that. I knew you guys would have it all. I'm just reinventing the wheel in the lab. Oh, that's interesting. Kelsey, this is probably a question for all of your colleagues here, not necessarily me, but yeah, I wonder how stressful it would be to isolate captive bred in order to carefully collect their molts, for example, or video them. I know they tend to, from what I've heard, they tend to crawl away and hide when they do this. And as you would expect, they're vulnerable. And so they don't want to have another crab come across them, which is very, very likely to abuse them or eat them or something. But if they're isolated, they may feel comfortable enough to do that. You guys have a much better sense of what sort of stress that would cause them than I do. Oh, very good. Yeah. Can you show us how to hold a live pinchy crab and measure its bits, please? I have held live pinchy land hermit crabs about 4 times in my life, Stacy. So, like I said, you guys have all the experience in this. They are pinchy, and they are difficult as they get bigger. They're a lot more difficult to handle. All those measurements that we did were very easily done on dead animals. It is a lot harder to do on live ones that are wiggling around. I suspect you should be able to get shell length fairly easily. The shield length, the carapace length from eyes to the back of that cephalic shield. If you had some sort of calipers or rule, you should be able to get a rough guide there. It gets harder and harder. You have to get them out of their shell to do the other measurements. Just thinking on my feet here. You could try putting something soft like cloth on them, so that would occupy all of their claws and legs, and leaving their abdomen and fifth legs exposed to get a quick idea of the width of or length of sexual tubes or coxal segments. And getting that one leg you want out could be a little challenging, but you might try something similar. I'm thinking about the way you might handle a cat if you wanted to clip its claws and it was non-responsive. All right, so you wrap it up in a towel, you grab that one leg, you poke it out, and while it's struggling inside the towel with its claws and teeth gnashing away, you clip the claws you want. So I'm thinking about keeping all of those legs and claws busy, maybe with some fabric, some soft fabric, nothing that's they can pull the fabric out of, so nothing so fluffy like cotton wool or anything, but a fabric that they can keep themselves busy pinching and scratching on while you're trying to get that one limb out. And like I said, if it's going to be too stressful, I don't want you guys terminating crabs just for that. But you might think about how you might go about doing that. You can slow them down by cooling them down. You don't have to freeze them, but you can put them in the fridge for a little while. 10 or 15 minutes in the refrigerator. They're tropical crabs. They won't like it, but it might slow them down a little bit. Think about that. So that could be a possibility as well. But once again, I wouldn't want you to threaten the health or lives of your crabs in that regard. I have Brooke, a one-eyed crab that knows a specific crab that has never grown it back. See, that's interesting. That's a great key marker for an individual, but I bet that's rare. But if you have crabs that have particular things like that, most structures that they lose, they're going to grow back, but some they may not. And so you may have structures that you can track over time. I think we're still talking about... Oh, that's interesting, Brooke. I'm looking at your comment a few minutes ago about people with crabs that are 8 years old and aren't very much bigger. Yeah, that would be a proof of concept of the fact that they might not be in ideal conditions. So if they're just surviving with a limited set of shell resources and they are surviving but not thriving, then they may not have the ability to grow very much. But you guys know very well what they need, and if you provide them with all of their resources in excess and extra shells, and as long as you don't have a too dense population, that might also be an issue. Population density, that would be an interesting thing to track from a research perspective as well. If you have a set area of your tank and you can manipulate population density by raising them in low density and high density, you may be able to find out if higher population densities restrict growth or not. It may increase interactions between crabs. They may be able to swap shells more, and it may not be a factor, but it may be a stress factor. Increased population density may increase stress, which may decrease molts and growth. So that's something you may be able to test as well. For those of you who have extra tanks and lots of crabs, you might think about doing something like that. Oh, interesting comment there, Judith, about if they do live over 40 years. Theoretically, yes. You should be able to keep providing them with larger and larger shells, and they should move into them. They would have to have other unlimited resources like food and have all of the minerals and be as healthy as they can to grow to that sort of size. Whether they would get as big as a coconut crab, I'm not sure. There might be some limitations, but theoretically, they should continue to grow. I've seen Maine lobsters in the Smithsonian's pickled collections that are 4 feet long from tail to claw. Okay, now that's not your regular size, but that is about 3-4 times the size of your usual plate commercial lobster. So that's a pretty impressive animal, and we have no idea how old it is. It will be reasonably old because to get to that size. But theoretically, with increasing shells and a long time and with all the resources it needs, a crab should continue to grow. I think it'll only be resource limited once it gets over a certain size. All right, Mary, looking at your comment here. We don't yet suspect hybridization. We have seen some strains captured. Oh, I just read that one. That one popped to the top. All right. Carbon dating won't work too for age counting. Carbon dating, things have to be hundreds to thousands of years old for carbon dating to really work. As if I'm thinking about carbon dating from like an archaeological perspective, things that are just dozens or years old, it doesn't really work for. And it works good. Carbon dating works from about 2,000-3,000 to about 50,000. Then after that, you have to go to other sorts of dating when I'm thinking of geological and archaeological stuff. Now, people have tried all sorts of things, and so far they haven't found a successful method for getting an accurate date, age of any commercial crab. And it's mostly been done on commercial crabs because the industry wants to know and pays the money to find out. So... Oh, I'm looking at Kelsey's comment there. I wonder if it could be socially learned. What are we talking about there, Kelsey? I'd have to go back and find out what you were talking to Mary about. Whether they're with older crabs or with babies. That's interesting. Yeah, that's something you guys can test too. You can raise crabs in a cohort all the same age, or you could put them into a mixed population, which would be more natural. They would be recruiting two populations back onto beaches with mixed population sizes in. That would be more natural, but it would be interesting to see if they're all the same age and raised as a cohort, whether that influences growth rate, or any other sorts of behaviors. It's quite interesting. All right, looking at something from Laura Carroll here. Larger tapestry turbos. Oh, that's interesting. Well, I'm sure they have preferences. I mean, that could be just the right size, just the right fit, and the turbo shell is the one they really want. The hardest thing is going to be accessing a whole lot of shells of the same species of different sizes. And I guess you guys get your shells from, what, do you go to the beach to get them? Do you buy them from Bed Bath & Beyond? I have no idea. Where do you get all your empty shells from? Do pet shops supply them? You'd have to be getting the same species and get a range of sizes. So that will be a task unto itself to be able to collect all that data. Yeah, finding bigger shells is a challenge, and you have to often go to look for commercial places that are selling shells to find shells that are big enough to fit some of your crabs. And that's why in some places I mentioned I've seen articles about crabs, land hermit crabs having their butts in the lips of broken beer bottles, plastic soda caps, and things like that, because they just not really accessing the shells they need. Yeah, absolutely. Yeah. Oh my God, that time went quickly. We just went through 50 minutes in no time at all. Yeah, I'd like to see Tammy's too. So can we all just bug out? And how long is Tammy's? Or maybe you want to send a email to me, Mary, or something, and let me know when everyone's gathering again. I'll watch Tammy's anyway, so you can let everyone know if we want to get back and talk about this. I have no trouble at all with that. We'll have to come back to this point if someone can remember where we're at after this. So do you want to, we're all going to bug out now and go set ourselves up to listen to Tammy's talk, I presume. All right. See you all back here in a little while then.